Our next presentation will be the U.S. Food and Drug Administration's Total Diet Study by Judith Sprungen and Terry Consul. Judith has a degree in nutrition and has been participating in various international bodies related to dietary exposure as well as risk assessment. Terry, are you presenting too?
Yes.
Okay. Terry, you started out as a geologist and now you're working for the FDA.
Yes, sir. Yes.
Oh, okay, the floor is yours then.
All right. Thank you so much, sir. Welcome from New York State, not the city. My colleague Judy Spunga is going to tag team this with us today, and she's out in Portland, Oregon, across on the west coast of the United States, 3 time zones away, so it's rather early for Judy. Thank you for being with us, Judy. So having a country that spans the entire continent brings forth a whole set of challenges for total diet studies, and we've modernized our program, so I hope to tell you a little bit about that. The U.S. Total Diet Study was initiated back in 1961 and has been conducted continuously. Many programs are sporadic and they focus on a certain set of analytes or a certain target population or foods. Ours has been conducted continuously since 1961. It's an important part of FDA, our Total Diet Study is in the Food and Drug Administration, Food and Nutrition Monitoring Program. And we focused mainly on pesticides, elements, radionuclides, and mycotoxins. And we determined these levels of analytes in foods as they would be consumed, table ready, as most of you do as well. Again, it started in 1961. The first total diet study looked at radionuclides and some pesticides and vitamins, and they took the composite of food, made a single composite, and analyzed it. Then they decided that that wasn't such a good idea, and they focused on different food groups. So they did the dairy, the breads, the meats separately, and fruits and vegetables. And that worked out much better. And then we started adding infant and toddler foods. And then in 1982, we analyzed each of the foods individually. In 91, we went to quarterly collections for all the foods. And in 2003, we updated the food list. And then in 2013, we realized that there were some problems with the way we were doing things. And we started the modernization process. And in 2018, we began our modernization, we implemented the plan. Total diet studies are not stagnant programs. It's a kind of a contradiction between wanting to keep things consistent, but wanting to make changes to keep up with modern technology, instrumentation, sampling, consumption of foods. And so it's a balancing act. And we decided in 2013 that we needed to modernize our program. So let me tell you a little bit about how we did that. All right, so our goal was to enhance our data quality and reduce uncertainty. And we did that through data management, our methods and data quality standards. We evaluated how our sampling plan was, and we hope to improve our food list mapping. And the key to all this was they did not give us an increased budget. As many of you pointed out, many of your budgets are on the lower side. The United States, I feel good that we have a good budget for analyzing food and conducting a total diet study, but we always want more. So for the data management, we needed a central database that we could store all the data that we collect. And it's a lot because we look at pesticides, elements, radionuclides, and mycotoxins, and we need to keep adding new analytes. We need to be able to retrieve that information fast and then do a lot of data review. We always do a lot of checks and everything. It used to take a significant amount of time, and we figured we could automate a lot of that, especially in the data reconciliation, and make things go faster. And then we added some graphics and visualization because that always helps when you're presenting data. Our methods, we updated to multi-residue and multi-analyte techniques for the most part back in 2017. It makes it a lot more cost efficient to do that. We have a standardized protocol now for determining LODs and LOQs. because FDA is the regulatory agency, and a lot of their methods that which we use are set for much higher levels of looking at regulatory limits. We need to look at those much lower levels. And then the way we do data analysis, handling lots of data sets with many non-detects, such as our South Korean friends just told us about. So we analyzed for 25 elements, including speciating for arsenic, as many of you do as well, on select foods and beverages. If it crosses a certain threshold, then we speciate. We look at about 300 pesticides, including glyphosate. We target 3 radionuclides that are major fission byproducts. However, on our gamma spectrum, if there's anything there, we always looked at the full spectrum. We would report it out. And then we started analyzing mycotoxins this year, and it's a new analyte for TDS. They came out with that new mycotoxin method that uses isotope dilution, and it became economically feasible mycotoxins before they were all single analyte, at least in the United States, FDA. There. We evaluated our sampling plan. We looked at how things were done between the 1991 and 2017. It was done consistently for that time frame, but it didn't capture a lot of things that we wanted. And so we wanted something that would capture seasonal variations throughout the year. Difference, again, we span a whole continent, and so there's a lot of different growing regions in the United States that are vastly different. The food list we needed to update, and we also changed it to some things would vary regionally, others would not vary so much, and so those were national and regional foods. And then we wanted a population-based sampling versus convenience sampling. So that study design that was done between 91 and 2017 basically divided the US into 4 regions. They were not done on equal population. That was not done on regional biologic or ecologic boundaries. They were basically FDA administrative boundaries. And so they didn't have a whole lot of meaning. The food items that were collected were from each region. They were sampled quarterly, but the same season each year when they did it. They didn't vary the seasons. And so we couldn't tell seasonality, we couldn't tell regionality really. And so that kind of didn't help us for any of that. TDS food was purchased each quarter. They went to supermarkets, and the foods were prepared table ready as they currently are. However, the selection of food retailers was at the discretion of the FDA staff. And what they would do was pick the one that was closest either to their house that they had to go to, or if they went to the office first, they would go to their office and pick the grocery store that was closest, that could get almost all the foods from, that was closest to the office. And each of the collections was called a market basket. And here's the 4 regions that they collected in. And then they collected from 3 cities in each of those 4 regions. The 3 cities were sent to the lab. This lab then would composite them, cook them, process them, and analyze. And then each of the samples would go mostly to the Kansas City lab for pesticides, elements, and nutritional elements. And they would go to the WEAC laboratory up in Massachusetts for radionuclides. So we decided to make a big change in our sampling. We would increase from 4 to 6 regions. One market basket would encompass 2 years worth of data that we would do the exposure analysis on. We wanted to make it population-based, so it'd be probability proportional to size. And we would break the food up into regional and national list. Regional foods were expected to have environmental differences that would be affected by environmental factors. California is hot and dry. California is very humid and wet and prone to thunderstorms and hurricanes and other things. And so the different pest pressures, different analytes in the rock and soil and water. And so there would be some differences. These would be mostly the breads, the milks, the meats. National foods would be mostly your processed foods. And then we'd include seasonality. So we would vary each of the regions throughout the year such that we could make seasonal determinations. And these are the regions. We tried to do them approximately equal population. We could have done it by ecological regions, or we could have done it by political social boundaries. We chose to do equal population. They're approximately equal population. We didn't want to sit there and cut counties out of states in order to make them exact. So this works within the boundaries and error. And we are including Alaska and Hawaii, which was not included in the past. So there's the 6 regions. They range between 50 and 55 million. Within each region, we sort the counties based on USDA has this urban influence codes that they came up with, National Statistic Service for USDA. And what we do is the more populated an area is, the more likely it'll be sampled. And the more rural areas will be sampled far less likely or chosen for sampling. And we select 6 counties within a region. 3 will be in the summer, 3 will be in winter. These are examples of USDA's UIC codes. The 1 and 2 would be sampled far more frequently than any of the others. So once we select a county that has a high population, what we do is we go to the US Postal Service, and they have a database full of all the addresses, and we filter out all the business addresses, and we take just residential addresses, and we randomly select a household. Then we go to Google Maps, and we find that address, and we put in to get the local retail grocery stores in the area. And we select grocery stores then from 6 food retailers that are near that location. And those are the ones that our FDA field officer will then go out and collect samples at. And here's an example showing Google Maps the address approximately in the grocery stores that are surrounding it. And then again, we collect samples twice a year, once in winter, once in each region for each season. And this is just a map showing kind of the population centers. If you know the United States, you can see that the Los Angeles has been sampled in San Francisco areas, and Seattle and Portland frequently, and the DC area, and New York City obviously, and in Florida, the big cities as well, frequently. So our current market baskets from 2018 through present, they consist of 24 regional collections, so 6 regions, 2 seasons each for 2 years. There are 93 regional foods collected at 3 cities in each of those regions, and then it's composited. And then we have 2 national collections. They are about 172 food items. These are the processed foods for the most part. And many times we tried to, each of the national foods needed to be encompass about 50% of the market share. And so sometimes we had to have multiple brands in order to do that, to make up 50% of the market share. We also vary the time of year that the national foods are collected. Being that it is a national food, it's usually made in 1 or 2 plants and distributed across the United States. We don't expect variations, so you could collect that sample, the national sample, at any location. And so we chose next to our laboratory to save shipping costs. In fact, our laboratory staff go out there and do the sample collection for the national collection, and it makes life easier. And again, we collect from 3 cities in the region. The 3 cities send their labs or samples to the lab, which are then processed, cooked, and analyzed there. And the national samples, the brands are composited and sent for analysis. And it's the same laboratory, our Kansas lab, doing the elements, radionuclides, and pesticides, and the WEAC laboratory still does radionuclides, so that has not changed. Based on the 2013-2014 NHANES consumption surveys, we've updated our food list. It's more specific now. We don't just say red apple, we give them some varieties. We based it a lot on sales data, such as the Nielsen or IRI sales data, and Judy will go into more of that. And then lastly, we put out, being that COVID hit, and we missed samples when the initial days of COVID. And so in order to encompass about 24 samples and get enough to do the report and exposure assessments, we had to include 2020. And so even though it's 3 years, we missed a lot of samples in that time frame. So we stopped at 2020 and we started looking at the data and put it up on the web and writing a report. So we do have a report that just came out for the elements data, and our web pages are now updated. We're working on the pesticides and the radionuclide data and getting that up on the web and writing reports for that. We like to write a report now such that it kind of explains the data a little bit and gives some context to it. In this, you will notice that there's a special study. TDS is starting to do some special studies in the United States. I'll explain in a minute. This particular one is baby food, so there's a lot of extra baby food samples in here for this particular report. In the United States, a lot of people have immigrated from all over the world to the United States, and so the diets, like to eat their own native and ethnic foods that they have. And so we have a special project ongoing now looking at some of these ethnic foods because they're consumed fairly highly, but not enough to get into our national list or our regional list. And so we have special studies looking at some of these things now, not just our normal production. The United States sits on about 30% of the world's arable land, and so we import a lot of different foods for these ethnic things that we don't have and grow in the United States. We also export a lot of food too, because we produce far more than what we consume. And so US total diet study may be important to your country because it might be coming from the United States, your food. And with that, I'm going to turn over the patient to my colleague, Judy Spungen, and she will tell you more about exposure assessments in the US. Thank you.
Okay, thank you, Terry. I'm just going to be speaking a bit about exposure assessments based on TDS data. Here are some examples of TDS-based exposure assessments we've published in the last few years. These have focused on perchlorate, iodine, lead, and cadmium. The results are based on individual consumption data using individual body weights, not reference diets. I'm not going to show the results because these papers are in the literature and they're freely available. They're not so recent anymore. We're working on updating these and other exposure assessments using our newly released TDS data and some updated methodology that I'll talk about. And I want to mention that although we do include nutrient elements in our TDS, that's mainly for monitoring purposes. Estimating intakes of those nutrients falls within the domain of the U.S. Department of Agriculture, not the Food and Drug Administration. Iodine is kind of an exception to that, and I'll talk a bit about iodine near the end of this talk. Our TDS-based exposure assessments really focus on contaminants. So instead, I'm going to talk about two exposure-related changes to the US TDS program. One is on how we treat non-detects, and one is how we're mapping the TDS constituent data to consumption data. So first, looking at what we do with non-detects, there are a lot of options. Everybody in this audience knows that. Historically, our TDS has reported lower exposure assessments based on lower bound means. Sometimes we have also included upper bound means. We're starting to use a hybrid mean, and Chowiel referred to this as the US FDA hybrid approach. We actually refer to it as the US EPA, Environmental Protection Agency approach, because they actually were the first in the US to do this for lead analysis. And what we do is assign non-detects to 0 for foods that we just have a historical history of no detections. And then we assign a middle bound, we assign half the LOD for samples with some history of detects. So just briefly, that's our new approach, that we're reporting lower exposures based on lower bound, upper bound, and hybrid. A really major change that we're working on right now is the way that we map TDS constituent data to consumption data. So our food consumption data source is the National Health and Nutrition Examination Surveys, or NHANES. And the dietary portion of that is called What We Eat in America. It's a funny name for a food consumption survey, but that's the name of it. I'll just call it NHANES What We Eat in America. And the current version of NHANES is fielded for 2-year cycles. The dietary portion since 2003 has consisted of 2 24-hour recalls conducted 3 to 10 days apart, and about 8,000 respondents for every cycle. There are well over 10,000, that should be 10,000 there, well over 10,000 different food codes reported to date. And USDA doesn't have nutrient data on all these 10,000 foods. So nutrient concentrations in each food have either been measured or estimated using recipes from a database called the USDA Food and Nutrient Database for Dietary Studies, and I'll refer to that as FNDDS. Going back to TDS, our TDS mapping through 2017 was not based on recipes at all. Instead, each of our foods was mapped to a NHANES What We Eat in America food. And the mapping was based on the major ingredient, not on packaging or any other attribute. And the values for each TDS index food were assumed to apply to all of the NHANES What We Eat in America foods to which the TDS food is mapped. So what does that mean? Here's an example. We had a TDS food, avocado raw. We still have that food. And we use the values for raw avocados not only to apply to intakes of raw avocado, but also to guacamole with tomatoes and guacamole with tomatoes and chili peppers. The problem with that is that we know that different pesticides are used on avocados than are used on tomatoes and chili peppers. Here's another example, boiled shrimp. Shrimp is the only shellfish that we analyze. It's the most highly consumed shellfish species in the U.S. And we have mapped the values for shrimp to other forms of shellfish, and we're still going to do that. But we're mapping values until 2018. We were mapping values for broiled shrimp to deviled crab, and deviled crab includes all kinds of other ingredients that should be considered. Here's our most famous example is until 2018, we sampled the ingredients for tuna noodle casserole, and our lab prepared a tuna noodle casserole homemade. Once upon a time, tuna noodle casserole was a very popular dish in the US. And it was chosen to represent all seafood starch food mixtures that were consumed in the food consumption surveys. What's the problem with this? We know that tuna has substantial concentrations of methylmercury, and the other foods to which it was mapped, such as flounder and just generic fish, may not have such high concentrations of methylmercury. The other issue is that tuna noodle casserole values were mapped to mixtures that contained rice, and we know that rice has high concentrations of inorganic arsenic, so we would be missing out on those by using the values for the tuna noodle casserole. So we have developed a new mapping approach for food mixtures. We have in the US these USDA recipes that are developed for nutrient values, and our thought was that we could build on that and estimate our contaminant concentration. So before 2018, we collected and analyzed egg, cheese, and ham on English muffin, lasagna with meat, chicken fillet sandwiches, tuna noodle casserole that I mentioned before. Our idea was that we would map based on percentages of the ingredients that we already collected for TDS. So going back to the FNDDS and the available recipes, here's an example of a food that for which USDA estimated nutrient contents based on recipes. It's a potato salad. TDS used to sample potato salad as well. And here are the nutrient values that USDA came up with based on a recipe for potato salad. And here's the actual recipe that was used for potato salad. It's a generic recipe. It may not apply to all potato salads, but it was chosen as a representative recipe. Here, I'll present a couple examples of how we'll use these recipes to estimate contaminant concentrations based on TDS data. So on the top, there's a recipe for cheese sandwich, not further specified. It's 41% cheese, 59% white bread. And we analyze an analogous cheese for TDS, and we analyze white bread. So that's an easy map. Cheese and bread using the same percentages. Here's one that's a little bit more iffy, but we feel pretty confident about it. This is for a gyro sandwich. It used to be, I should mention that cheese sandwich used to be mapped to macaroni and cheese because that was the only starch cheese mixture that we had in TDS, and mapping a cheese sandwich to macaroni and cheese is not ideal. It used to be that this gyro sandwich was mapped to a hamburger because it's a meat and various starch ingredients. We're using the FNDDS recipe on the left and the analogous TDS ingredients on the right to come up with contaminant concentrations for this gyro sandwich. We don't have an exact match for the Greek yogurt or the pita bread, but we have ingredients that are close enough, and we can do some moisture adjustments where things aren't exact. We have a situation where some of the USDA foods, if they had nutrient data for the food, they didn't develop a recipe. So we had to develop some of our own recipes. This is an example. This egg, cheese, and ham on English muffin. This is a popular fast food item. And USDA analyzed this for nutrient concentrations. We developed a recipe based on known ingredients. We weren't sure in this whether to include margarine or salt. So we looked at duplicating the nutrient contents and seeing how close we could come with a recipe. The orange line is what we calculated. The green line is what USDA reported. We were already over in fat without including any margarine, so we just decided we're not going to include margarine in that recipe. So just to summarize the changes that Terry discussed and the mapping that I discussed, we went from a 4 market basket collections per 1 market basket every 2 years. And in this current weird Corona situation, it was 3 years. From 4 regions, from 1 food list to 2 food lists, a national. We went from convenience-based sampling to population-based sampling and a much more specific criteria for shopping and obtaining samples. And we have revised our treatment of non-detects. That's not on this chart, and then we're looking at this new mapping system that I described. I said that I would talk a little bit about iodine. Iodine is a nutrient and a contaminant, as we know. USDA has not typically analyzed for iodine, but they started analyzing for iodine. So we have a collaborative project with USDA, the NIH Office of Dietary Supplements, and the Centers for Disease Control, combining USDA's data on iodine concentrations and the TDS data on iodine concentrations. We're using a mapping system similar to what I described. It's a little bit different because we also have the foods that USDA analyzed. And we're going to compare our exposure estimated using the mapping system with urinary iodine concentration data for a subsampling respondents. Again, just we TDS data use are as important part of a food safety program. The TDS data are very, very critical for our toxic elements working group and doing policy based on the findings. We have a action plan called Closer to Zero. There's been concern by the public about exposures to young children, and we're using these data and other data developed by FDA to reduce, as far as we can, the levels of arsenic, lead, cadmium, and mercury in foods, especially foods consumed by children. We're setting some action levels with input from stakeholders. We're looking at industry practices to hopefully lower the levels of toxic elements in raw agricultural commodities. We're doing increased compliance and enforcement, and using the TDS data and other data to monitor progress in levels of these contaminants over time. We have a website. We have an email address. Please feel free to write to us. Just some acknowledgments. We have a team of 7 and we have a much larger team, but the people who administer and do the analyses of the data that are produced, there's 7 of us. None of us, actually, Terry works on TDS full-time. The rest of us work on TDS part-time. We also work on other FDA initiatives and activities. Our Kansas City laboratory personnel do so much work for us, and we have sample collectors out in the field. Our TDS is a huge effort, and we thank and acknowledge all these contributors. And now Terry and I would be happy to take questions.